So I'm Fernando Vieira. I'm the Chief Scientific Officer here at ALS TDI. And I'm going to be telling you today about our comprehensive research strategy to end ALS. Comprehensive is kind of an interesting word. And I'm giving a comprehensive, I'm talking about the comprehensive strategy, but I'm not giving you a comprehensive outlook of the work that we do. As I was thinking about giving this presentation today, I was actually pretty overwhelmed by the amount of work that we've gotten done in the past year and that we kind of always get done. So I'm going to give a relatively surface overview of our program, hitting specific highlights along the way. But I'm hoping that by the end of the day, you'll have an opportunity to get more depth by talking to other members of the science team here at ALS TDI. So before I tell you sort of about what we do, and perhaps more importantly, why we do it, I want to tell you a little bit about who we are. So ALS Therapy Development Institute is a pretty unique nonprofit organization. We're a place where we hire our own scientists, our own staff. We execute our own experiments in this laboratory, ALS Therapy Development Institute, in the heart of Kendall Square. And we have all of these wonderful people who do that work. And the reason I kind of want you to get used to this sort of schematic, because I'm going to use it through the presentation. As I'm going through the highlights of work that we're doing at the lab, you're going to see pictures of individuals who are kind of doing that work, and you're going to kind of see the sections of the lab space that get highlighted where that work gets done. And I'm doing that for a specific reason. I kind of want you to get a sense of how it works for all of us every day when we're going to work to try to end ALS. We talk a lot about the idea of collaboration, and collaboration is really, really important. We collaborate with a lot of different organizations, ALS-related organizations, organizations that didn't have anything to do with ALS before. I mean, we've got a bunch of folks from Google here today who are some of our favorite collaborators. So we do try to find ways to work with lots of different folks to leverage our organization's expertise and their expertise for the best outcomes for people with ALS. But there's also sort of a different type of collaboration that occurs when you all go to the same place every day and are kind of tripping over each other in the halls. And we have we literally do trip over each other. We're clumsy people, but not while doing experiments and pipetting. And so we have multidisciplinary folks, people with lots of different skill sets and backgrounds. Everything from genetics, genomics, pharmacology. We look at clinical endpoint measures. We have cell-based assays. There's a lot that we bring to bear, and it's all happening in this location, and we're all bringing our perspectives. So I kind of want to hammer that home. And that's a theme for the day. We're doing something new today. So you're hearing from me. You're also going to hear from Dr. Theo Hatzapetros today about our pharmacology programs. He's going to speak right after me. And then we are going to have something new that we've never done before. We're doing poster sessions. So a lot of the folks that you're seeing here, they'll show up. The ones who are doing posters will show up kind of highlighted in purple. I'm trying to stay on brand. And they'll be presenting posters that can give you a little bit more of a sense of depth on the specific projects that I'm talking about. Is this my advancer? Okay. No. Middle button? Thanks, Theo. All right. So last year, I presented to you a framework of the obstacles that we all confront when we're trying to end ALS. And there are a lot of different ways that you could frame the obstacles to solving this problem, but we have a certain framework that we kind of lay over the problem. And I'm going to walk through those like I did last year, and then I'm going to walk through the strategies that we employ to try to confront and overcome those obstacles. And then I'm going to tell you in more detail about some of the progress that we've made against those strategies. So one of the first things that we need to address is that we still need more good quality drugs for ALS clinical trials. We need to enrich the clinical trial pipeline for ALS. And I know things actually look pretty good these days compared to how they were, 10, 15, 20 years ago. But we still have fewer than 30 drugs in the ALS clinical pipeline. So that means that there are fewer than 30 possible drugs that are in phase 1, 2, or 3 clinical trials for ALS right now. And all right, that sounds good, but for context, in 2017, in Alzheimer's, there were just shy of 130 drugs that were in clinical trial. So that was 2017, and now we're on the verge of 2020, and none of those drugs have actually been approved yet, right? And Alzheimer's is not a more complicated or difficult to solve disease than ALS. But it does get more investment, right? And there are more options available in clinical trial right now. So that's a problem that we need to address. Another problem that we need to address is the complexity of ALS. Dr. Perrin alluded to it in his presentation. He said ALS is complex. He gave you that massive slide with all known human pathophysiology that's been implicated. And that's true, and it's true on multiple levels. The biology of ALS is really complex, but so is the clinical presentation, right? So ALS, you have some folks who have really rapid disease progressions. We have some folks who have slower disease progressions. We have bulbar onset. We have limb onset. This heterogeneity makes it difficult to, number one, understand the disease. Number two, measure when you're actually intervening in the disease. So that's one additional problem. Another challenge that we all think about is that clinical trials are currently really inefficient for ALS. So even if we did enrich the clinical trial pipeline with more drugs, and even if we had a better understanding of the complexity and how to measure all of this, we couldn't actually get through all of those drugs in clinical trials because to measure drug effects, you need a lot of people. And there aren't that many people with ALS. That's a good thing. But it makes solving this problem really, really hard. Sorry, I got distracted. We need to find a solution to this problem as well. And then overarching on all of this, it's kind of a theme that sits on top of it all, is we really just don't know enough. I mean, obviously I said that ALS is complex, but I mean, when you look at ALS and you break it down like Steve did into familial and sporadic ALS, even when you focus just on familial ALS, there are more than 30 different genes that have been implicated in it. And of those genes, they all have lots of different functions. So just that 10% of ALS is incredibly diverse, and we have some insights into those, but the other 90% remain pretty much a black box. So we need to fill that knowledge gap. So now, what do we do at ALS TDI to try to address and overcome these obstacles? So, in order to enrich the clinical development pipeline, we screen drugs. And I'm going to tell you more about how we screen drugs and some of the progress that we've made in that domain. But it is a cornerstone of everything that we've done since our inception. To deal with the complexity of ALS, we try to create and use laboratory models that encompass all of that complexity. And I'm going to tell you some of the progress that we've made to address those challenges. To deal with the inefficiencies of clinical trials, we're working to discover and develop clinical outcome measures and biomarkers that could actually speed this process up. I'm going to tell you a little bit about that. And then again, overarching the whole thing, the knowledge gap, we're trying to learn about ALS from people with ALS. And that alludes to what Steve was saying, that we've now been able to bring people with ALS into this whole process. So I'm excited to kind of walk you through what we've been doing. So when I say we screen drugs for ALS, and we say this, it's in some of our marketing stuff, we say, we screen more drugs than anybody. Like, what does that even mean, right? Have you ever wondered? Because we say that, right? Okay. All right, so what is drug screening? So when you look it up, it's a method of scientific experimentation which is used to identify active compounds that modulate a biological pathway or disease process. So in some conditions, this is pretty easy to do, right? You might have a bacterial infection, and it is understood that essential for that bacteria's growth and its subsequent infection of you is an enzyme. And if you find a way to inhibit that enzyme, you can kill that bug. So you can develop a drug screen that kills that bug. But in other conditions, it can be a little bit trickier. So before I dive deeper into this, I am going to walk through a similar slide to what Steve showed. I'm not going to go into as much detail because he walked through it. But why I'm presenting this is to show you where drug screens sit in this pipeline, sort of in this clinical drug development pipeline. So typically, in basic research paradigms and academic labs, you'll find an interesting area of biology, and then that area of biology can be exploited to turn into a drug screen. So like I just alluded to with the example of like a bacteria, you could build a drug screen. And often those are done in cell-based assays, which are much more screenable. You can test in these cell-based assays, sometimes on the order of tens of thousands of molecules, even millions. Then you can advance those into other types of screens, which can be animal models, which are often a lot less screenable. They're less scalable, but they give you the opportunity to learn more about how a molecule is behaving, and then you kind of throw it over that wall into clinical development with phase 1, 2, and 3. So to show you a little bit about what we do at ALS TDI, for the majority of our time, we have conducted drug screens in animal models using SOD1 G93A mice. Just to remind you of this motif, these are the team members who work on this type of work, testing drugs in SOD1 G93A mice. And these are the areas of the lab where they do this work. So we've been doing this probably since about 2001. I think we're the best at it in the world, but we weren't born that way. We kind of got there by screwing up. Actually, we thought at first that we were really bad at it. And the reason we thought we were bad at it is because every time we tried to replicate somebody else's published result, we would get a different result. So these are examples of drugs that had been published by other groups to be efficacious in this SOD1 G93A mouse. I'm not going to go into a whole lot of detail about how this mouse works because Dr. Hatzapetros is going to, but the mouse gets ALS. The mouse has a gene that overexpresses a mutant protein that's known to cause ALS, and then it gets ALS and dies early. So when you work with this mouse, you try to deliver drugs to it and see if you can make it live a better and longer life. So other labs had been trying to do that, and they claimed efficacy, and we didn't. We weren't seeing it, and we were wondering why. And it turned out that it was because there were a lot of details that we were probably taking into account that other folks weren't at the time. Turns out you can't do it in only like 10 mice. You actually need to have enough mice in the study so that you don't fool yourself and accidentally get something that looks like a beneficial or negative effect. You also need to blind your study. The people who are observing the animals can't know which one is the treatment group and which is the control group. You also need to take into account which litters the animals came from. So mice that come from the same families tend to live to be about the same age. So there were all of these parameters that you need to take into account to avoid getting spurious effects. So we've been doing that for a while. For a long time, it was really frustrating because we weren't getting hits. Drugs weren't working. But more recently, our results and the results of the ALS field have been sort of more corroborated. I think the field has gotten better at doing these types of tests, and I think we're getting better at picking therapeutic drugs. And because of that, in the past few years, we've actually been able to validate 3 molecules in the SOD1 mice that have subsequently moved forward into human clinical trial. The first is our own homegrown AT-1501. It's an anti-CD40 ligand antibody that's been partnered with Analyxis Therapeutics that's now in phase 1 clinical trials in people with ALS. Another is a drug targeting misfolded SOD1 that we worked on in partnership with a company in Switzerland called Neuroimmune that has, about 4 weeks ago, was advanced into human clinical trials for ALS. And this third this wasn't a collaboration with the company in Australia. This was just our decision to try to replicate the work, but we did replicate copper ATSM as efficacious in SOD1 mice, and that has moved forward into phase 2/3 clinical trial in Australia. So this is really exciting. There's progress. A number of the drugs that have now come through our system are in humans. But something that we've always wanted to do was have a system in front of the mice. Remember? Here. A cell-based system that was more scalable. A system where we could test not, dozens of drugs per year, but a system where now you can test tens of thousands of drugs per year. And we sort of struggled over this. We struggled with what would be a relevant assay system, what would recapitulate any features of ALS. And thanks to progress from the translational research team that was born out of the precision medicine program that Steve alluded to, we've made advancements there. In the past year, we've now established a drug screen which uses human ALS-related induced pluripotent stem cells. So this is really exciting because now, instead of simply reading papers in PubMed and kind of taking some shots and some guesses and saying, oh, we think this might work in the mouse. Now we can take hundreds, thousands, tens of thousands of drugs, test them up front in cells, and then find the molecules that are hits, and those hits then have a better chance of being efficacious in the SOD1 mouse. So we're really excited about that. And this is the team, the translational research team that has worked very hard to develop these models, and that's the space they work in. And Dr. Kyle Denton will be presenting a poster today speaking in more detail about how we use induced pluripotent stem cells as a drug screen ahead of our mouse screen for ALS. In brief, I won't go over this in too much detail. If you don't know what an induced pluripotent stem cell is, somebody giggled. An induced pluripotent stem cell is a cell that you can derive from an adult. We tend to get them from a skin punch, a skin biopsy. You can culture fibroblasts out of that skin biopsy. And then you can add a few genes to those cells, which coaxes them into becoming any type of cell. So they have a pluripotency. They can become any type of cell. Once they're pluripotent, you can then put them into different protein milieus or growth factor milieus and coax them into becoming, say, a motor neuron or an astrocyte or back to a fibroblast. There are lots of different ways you can measure differences between an iPSC-derived cell from a person with ALS or an ALS-related mutation and compare them to healthy. If you see differences in the ALS-associated cells compared to healthy, then you can look for drugs that rescue those differences, and that's how the drug screen works. So we're really excited about this advance in our lab. So to summarize, as far as how we screen drugs, remember this is to enrich the clinical development pipeline. ALS TDI has established a drug discovery screening platform for using induced pluripotent stem cells and an animal model of ALS to identify promising drugs for clinical development. So now we move on to trying to address the complexity of ALS using models. So back to the question, what is a model? You hear us use this word a lot. So you've now heard me talk about two different models. You've heard me talk about the SOD1 G93A mouse, and you've also heard me talk about induced pluripotent stem cell-based screening models. But what is a model? It's a system. A model system is an organism or another self-contained system that is used to evaluate a particular biological activity or disease process. We have a lot of folks at ALS TDI that are working hard to develop these types of models, and I'm going to kind of try to explain how we think about models a little bit before I dive in. So this is the way I think about them. I think about models on a spectrum of relevance to people living with ALS on one end, which are maybe less scalable, and more scalable models, which might be less relevant. So let me walk through that. What would be the most relevant model of human ALS? Well, it would be a human with ALS. But let's think about that for a second. We all know that there are lots of different types of ALS, right? So there's no one person with ALS who would accurately recapitulate another person with ALS. A man with ALS does not model a woman with ALS. A SOD1 patient with ALS doesn't model a C9orf72 patient with ALS. There is no one model that is that would be appropriate to cover all. So let's dispense of the idea that we can ever have like a best model. What that means is we have to have multiple models. So let's walk through the spectrum of models. So on this other end, so here we have the most relevant and the least scalable. But on this end, we have the most scalable, but right now, potentially the least relevant. So these are in silico models. What this means is it's working in a computer system. You feed a computer system. We're hoping this gets better, Google. Right now, you would feed a system with data, and then you would use those data to predict outcomes without ever actually having to do a wet lab experiment or a clinical trial. Right now, so you could do this all day, right? You could just plug this into a computer and let it go. But right now, these aren't particularly predictive of outcomes in humans. And then you get to a little bit more relevant model. You might have an in vitro model. If you want to focus on like a specific enzyme, you mix that enzyme up into a tube, and then you ask about that enzyme's activity. An enzyme is usually doing a job in a cell. And you might try to find drugs that block that activity. This is pretty easy to do, pretty scalable. Again, not necessarily relevant if that enzyme has nothing to do with your disease. Then you get to a more complex system. You can now work in cells. So cells are still pretty easy to work with. You can scale them, like I said earlier, using cell models. You can test tens of thousands of compounds, sometimes even millions. But you're still pretty far away from a human, even if they're human cells, right? So what you're usually talking about is a cell system, a single cell type, working in isolation in a plastic dish. You can be studying motor neurons, which is really relevant, but you're not studying their interaction with muscles, or you're not studying the oligodendrocytes that are producing the myelin that wraps those axons. The whole complex organism is very difficult to model in a dish. Then, much, much closer to humans are non-human mammals, right? So mice. So we do a lot of work in mice. They do model that more complex system. You're not working with human cells. You might not be working with a human genome, but there's a whole lot of overlap in those systems. And you can study things like side effects in a non-human mammal, right? So you might find a drug that's really effective on protecting motor neurons in a dish, but in that dish, you're never going to notice that it causes liver toxicity. If you move that molecule into a non-human mammal, that's the type of thing you have the opportunity to notice. And then, this is a bit of a spoiler, so we'll talk about this later. There are these sort of intermediate models, things like C. elegans worms, Drosophila, fruit flies, and things like zebrafish, which are kind of in the middle. Not quite a mammal, but definitely have complex tissues and organisms, and they're much more scalable than, say, working with mice. So having laid that sort of context for how we think about models, I'm going to talk about our quest to find alternate mouse models of ALS, aside from this SOD1G93A mouse model that I already described. So this is work largely spearheaded by Kaylee Mueller, and Matt Farola has taken on a role in this. Over the past year, we have tried to look closely at a lot of non-SOD1G93A mouse models. These top 4 here all represent attempted models of C9orf72 mediated ALS. Some of them behaved exactly the way we expected them to, in that they had no disease phenotype. We were hoping we might stumble into something different, but the reports said that we shouldn't expect a phenotype. One of them was expected to have a phenotype, but it isn't behaving in our hands, which is how things often work in science. You often see a paper get published and it's really, really exciting, and then something goes wrong between that paper and when you try to do the study yourself. Just kind of want to make that clear. And then we've also looked at a couple of different TDP43 related models. TDP43 is another gene which, when mutated, is known to cause ALS. And has been identified as a pathological hallmark in the spinal cords and brains of lots of people with ALS. But these mice, while they get a disease, it's not quite ALS. It's not quite the disease that we could use to study. And then finally, another mouse exploiting another gene which is known to cause ALS, profilin, did start to behave. And this seems like it's going to be a model that we can use for an alternate screen. So this is another type of model where if we're also testing drugs in SOD1G93A and we find efficacy there, we could test in this mouse and see if we're delaying hind limb or forelimb symptoms, paralysis, weight loss, and improving their lifespan. So all of this is to say that, that we now have a tool which will give us a better sense of the applicability of a drug that we're trying to move into the clinic. So I mentioned that C9orf72 kind of stumped us on the mouse model side of things. So that forced us to think sort of outside of the box, or outside of the cage, if you will. Come on, that's something. I'm a dad. We're now thinking in the tank, I guess. This is work. I know, I'm just going with it. Now you're all awake, though. So this is work spearheaded by Val Tassinari, and Ken Thompson did a whole lot of work on it too. Val, turns out she has a marine biology background, and it was something that we were able to take advantage of when we realized that we needed to find a new solution to the C9orf72 model problem. So I'll get to why in a moment, but just suffice to say that we have established 2 stable housing units for zebrafish, which is actually saying a lot. Val's going to be showing a poster today where she'll kind of show you the facilities that we've built. One of them was kind of done by hand, and another was one that we ended up sort of purchasing. I'm told the one that we built by hand works better, but we'll see. We've established a breeding protocol for these types of animals, so we can make baby fish at ALS TDI now. We've kind of dedicated this room to it. And we did establish a collaboration with a zebrafish neurodegeneration expert, Dr. Tanor Ramesh. So for those of you who have been around ALS TDI for a long time, you might know that he had my title before Steve Perrin or I did. So he was the chief scientific officer here in the early aughts. So it's kind of fun to go back and work with him again. But he has developed multiple models of ALS using zebrafish, one looking at SOD1, and he's also developed a couple of C9orf72 zebrafish models. So we're really excited to be taking steps in that direction where we can move to screen drugs targeting C9orf72. So in short, ALS TDI is working to deal with the complexity of ALS by employing and adding model systems to better understand the disease and to better reveal treatments that can be targeted to either specific subtypes of ALS, or we might learn that they're really broadly applicable. Something that works in the zebrafish, something that works in profilin mice, and something that works in SOD1 mice, that's now making you feel pretty confident that that's going to work for a lot of people with ALS. So that's how we try to address that problem. Now, clinical trials, clinical outcome measures. So I'm going to go on sort of a little tangent for a moment here before I dive into what we're doing with clinical trials, clinical outcome measures. Clinical trials are a big topic of conversation in the ALS space right now. This is something people care deeply about. There are important questions being asked and discussed around access to promising but unproven treatments. There are questions that are being asked about sort of the ethics of the application of placebo groups. And there are some innovations that are moving forward that are trying to address these. You know, we've all heard about the work being done at MGH Healy Center with platform trials. And this is a really interesting and important topic. We look at it from a little bit of a different angle because you have to remember who we are. We're not policy makers. We are not clinical trialists. We are a research institute, right? So we're scientists. So our way to address this is a little bit different. We're thinking about the problem, okay, so people want access to treatments. People want access to unproven treatments. We think, well, one way to speed up the whole process is to make it faster to learn if an unproven treatment is proven, right? So if we can change the entire paradigm by making it so that clinical trials could be faster, so that you can get your answer quicker, then that solves the problem, or at least partially solves the problem. So that's our angle. And so we're working to address that. And let me talk about that a little bit. Before I do that, I guess I should explain what a clinical outcome measure is. For those of you who don't know, it's a measure within medical practice or research which is used to assess the effect, both positive or negative, of an intervention or treatment. So in the case of ALS, current outcome measures can be really subjective and lack sensitivity to detect those changes. We often are using now something called ALS Functional Rating Scale, right? I talked about this in some detail last year. I'm not going to go into it in detail today, but I will say that what it is a pretty subjective outcome measure that asks people about how they can conduct activities in their daily lives. So everything from walking upstairs to turning in bed to being able to cut your own food or swallow or speak. These are not continuous types of data, so you kind of have to make a judgment call about when your multiple choice answer has dropped enough to warrant a single point dip. So our way of trying to address this is through ALS TDI's precision medicine program. Steve already talked about it a bit. We currently have 590 full participants who have shared with us, thank you to all of you who are in the program and who have shared data, who have shared at least 3 months worth of data. Many people have now shared years worth of data with the program. We've had more than 1,000 participants contributing data in general. We collect lots of different types of data in this program, but for this talk, I'm going to focus on clinical outcome measures. One of them, I just mentioned, is ALS Functional Rating Scale. Another is activity tracking using accelerometers. We have somebody in the room wearing 4 of them right now. And another is collecting speech voice recordings. So I'm going to start by talking about what we give back to the patients that give the data to us. We share it all back, like Steve said. This is a figure that a person who's participating in the precision medicine program would see if they log into their own portal. If you haven't seen these before, what you're seeing on the y-axis is ALS Functional Rating Scale plotted. That's this gray line here. For those of you on that side, it's that one, plotting the functional rating scale along the right axis. And then on the left y-axis, you're seeing vector magnitude. So those are the accelerometer data. So the accelerometer data are these lines plotted against this axis. You can see in this case, this person has their legs showing some decline. And then their arms are relatively stable for some time, and then they show a decline. And then this is a core measurement, which does not show the decline. So this is all to say that we really do work in complete partnership with our participants. We're grateful for the data that they share, and we want to give that back. But what we're trying to do with these types of outcome measures is have more sensitive and quantitative sense of when a therapeutic might be helping, right? So if a person is showing some decline along the way and then they start to use an intervention and then they see a bump up with their accelerometer scores, it informs both them and us when something might be promising. And in this case, I am kind of curious about what happened here with core. So blood-based biomarkers, though, also present a really big opportunity for ALS TDI or for the ALS research community in general. And if you don't know what a blood biomarker is, a really good example is glucose for diabetes, right? So it's a really clear example of being able to measure a physiological state by looking at a small blood sample and say, okay, now I need to deploy a treatment immediately or not. We lack this type of biomarker in ALS, but one of the reasons that the ALS community has had trouble identifying blood-based biomarkers or validating blood-based biomarkers is because we really haven't had high-resolution clinical data against which to correlate anything that we measure in blood. So we think that our high-resolution clinical outcome data using accelerometers and maybe even voice can reveal blood-based biomarkers that actually have a lot more potential. But we never had a way to measure blood. We never had a way to collect the blood, really, in our program. We're not a clinic. And it would be incredibly burdensome for people with ALS to try to come to ALS TDI to give us longitudinal blood draws. So we figured out, all right, how are we going to solve that problem? We need to collect blood samples to try to discover blood-based biomarkers, but we don't know how to do it. So our clinical team, spearheaded in large part by Alan Primaseri, who's also going to be presenting a poster on this today, built a system where we're working with a phlebotomy network, Phlebotomy, who will deploy and send phlebotomists to PMP participant homes to collect blood on a quarterly basis. So that's once every 3 months. The phlebotomist goes to your house. We will have already sent a box with tubes. It'll be sitting there. Phlebotomist draws the blood, processes the blood on site. They'll actually centrifuge the samples. They package those samples up, send them back to us at ALS TDI, and then we can analyze them lots of different ways. Everything from complete blood count to clinical chemistry to FACS analysis, flow cytometry, looking at multiple different RNAs or proteins. This really is a high potential program for us. So we've made some progress. We're aggressively exploring and validating clinical outcome measure technologies using accelerometers, looking at voice recordings, using machine learning, which Michael, Dr. Brenner from Google will be talking a little bit about later today, and blood-based biomarker discovery efforts to improve clinical trial efficiency and inform patient care. And then finally, we're still trying to bridge the entire knowledge gap in the ALS space. We still just don't know enough. And I've alluded to this through the entire presentation. We're really learning about ALS from people with ALS, especially through the precision medicine program. We figure that the best way to learn about hereditary forms of ALS is to collect material, cellular, blood material, biomarker material from people living with hereditary ALS. The best way to learn about environmental factors causing or driving ALS is to ask people about their environment, both before they had ALS and after they're living with ALS now. We figured the best way to learn about bulbar ALS is to listen, literally listen to people with bulbar ALS symptoms. So we're collecting voice recordings. And the best way to learn about slower ALS progression is probably to learn from people with slower ALS progressions. So I actually stole this slide out of my own presentation last year because it's the same. Our strategy remains the same. We aim to discover more ALS clinical trial candidates by screening drugs. We aim to address the complexity of ALS by validating and optimizing diverse models of the disease. We aim to produce better tools for use in clinical trials to speed up that process. And we aim to learn more about ALS by working with all of you. So back to the team. You've seen pictures of all of us through the slide presentation. And we always see pictures of all of you. This is just one of the walls at ALS TDI. They're all covered with pictures of you. It's often the case that, I'll kind of pop out my office and I'll find somebody who works at TDI stopped looking at a picture, reading the names, and thinking about their story. We're not in this fight alone. I've talked about us a lot, but we have a whole lot of collaborators through the years and now. So much of this is thanks to Augie's Quest and their funding efforts through the years. Thank you, Lynn, Shannon. We've had great partners along the way, including Google, who are here today, and lots of different collaborators. So thank you, and I will take questions if you're not tired of hearing me.